presenter. Thanks, Phil, for joining us. He spent his entire life living and working in the prairies. I look forward to learning more about your new role. So we've seen you in previous roles. So great to have you join us. Thanks so much.
Great. Thanks, Jamie, and thanks for the invitation. So I was invited today to talk about Envita, which is nitrogen-fixing bacteria, similar to rhizobia, and we'll get into that a little bit, but the big difference will be that it does work in all crops, and we'll kind of take a look at how that works here. So what I would like to do today is we'll take a little bit of a look at nitrogen fixation, specifically how it works, biological nitrogen fixation, and then a little bit more specific how something like Envita works, and then we'll go really a little bit more in depth into Envita, some of the science behind it, some of the results we're seeing to date, and as well as a performance guarantee we have for the farmers in the group. So the big benefit with a product like Envita, so if you're going to be providing nitrogen in the plant, fixing nitrogen in the plant, you're going to have that nitrogen in the right place at the right time. So the key with Envita is that it is systemic. So when you apply it, and we'll talk about application here, is it's going to get into the plant, colonize the plant, and then move throughout it. And that includes moving into new leaves. So most of the Envita gets applied with herbicide. So let's say 4, 5, 6 leaf stage. After that, as new leaves get produced, the bacteria will move into those new leaves as well. So it grows with the plant would be the way to think of it. And then really where we think we're seeing the best benefit or the benefit from Envita is later in the season. So currently we recommend keeping your nitrogen rates roughly where they're at, your fertilizer rates. And as that fertilizer gets used up by the plant or lost to the environment, the demand of the plant's going to go up because the plant is getting bigger. And then at that point, you're going to have the Envita in the plant fixing nitrogen, kind of feeding it directly to the plant as it needs it. And we'll take a look at how it does that here. So before we get into that, just a kind of a review here. So we are talking about bacterial nitrogen fixation. So it's similar to industrial nitrogen fixation or the Haber-Bosch process. So it converts atmospheric nitrogen into ammonia. Specifically with Envita, it does use the nitrogenase enzyme, which is the same enzyme that rhizobia uses. So you're used to using an inoculant with your pulses, and it's, you know, a specific rhizobia for a specific pulse. They all use the nitrogenase enzyme, and Envita uses that exact same enzyme. So that process is actually the same. There's just a few differences in terms of where it's located and how it's doing it specifically, but it's similar to what rhizobia does. In terms of nitrogen fixing bacteria, there's actually a number of different bacteria that do fix nitrogen. We touched on rhizobia and Envita. The big difference with products like Envita and then your rhizobia for pulses is they fix the nitrogen inside vesicles, and we'll take a look at what that means here specifically. But really, it's thinking of the nodule that rhizobia produces on the outside of the root. That would be the vesicle in terms of rhizobia. So it produces the nitrogen inside that vesicle, and Envita does something similar. There's also what are called free living in the rhizosphere. So there's bacteria that fix nitrogen just right next to the root. And so they'll take that atmospheric nitrogen, convert it to nitrogen, and make it available. But the plant is still responsible for sourcing it and pulling it in. And then there's some that are just free living in the soil. So that can be anywhere in the soil. It doesn't have to be in the rhizosphere. And those, again, would be fixing nitrogen, but making it available to the plant if it can source it. So benefits of symbiotic nitrogen fixation or nitrogen fixation inside a vesicle is that it's being fixed right inside the plant or made directly available to the plant. So you have no risk of nitrogen loss. It's made directly available. There's no competition from soil microorganisms or anything like that. And it's going to provide that consistent source of nitrogen, regardless of even what's going on in the soil in terms of type of soil or anything like that. So just quickly, Azotic is a company that is based in the UK as the head office. That's where the bacteria is fermented currently. The reason I throw this up here, though, is the bacteria was originally discovered in 1988. And so there's been a lot of research behind it, and it was launched commercially initially in the US in 2019. So there's over 30 years of research and development behind the bacteria so that we can demonstrate A, that it works, and B, that it's made in a form that is very user-friendly. So specifically, Envita is a naturally occurring food-grade bacteria. So the name of it is Gluconacetobacter diazotrophicus. As I mentioned, it was first isolated in 1988 from the sugarcane plant. So sugarcane was always known to fix a little bit of nitrogen. And so what they were able to determine was specifically which bacteria was doing it. And then once they discovered that, they picked a specific strain, and that strain that works in all crops and is easy to use. You can tank mix it with herbicide, all that kind of stuff was picked out. So that's what kind of happened over the 30 years between the isolation and the launch to commercialization. Quick product overview here. So in terms of Western Canada, it's registered really on all crops we grow in Western Canada. So canola, cereals, pulses, potatoes, for those of you in Southern Alberta. It's a liquid formulation, so it can either go on as an in-furrow or foliar. Most of it goes on as a foliar application because it's just so much easier. Very low use rate. So each of these jugs holds a gallon or 3.78 liters, and they'll each do 40 acres. It is a bacteria, so it can't freeze, and it has just a 1-year shelf life. For 2023, pricing is going to be about $16 an acre. And starting for 2023 as well, Syngenta Canada is going to be the distributor here for Envita. So your local Syngenta rep can help you out with Envita as well. So we'll take a look here a little bit on how Envita works. So first thing is, let's say this is showing a application with a herbicide, so as a foliar application. It's going to get into the plant. It'll either enter through the stomata if the stomata are open, or if they're not, there are some bacterial enzymes that will help the bacteria enter the plant. So we're not overly concerned if the stomata are open or closed necessarily for it to enter, but of course, if the stomata are open, that would be the path of least resistance. As I've touched on, once it enters the plant, it is systemic. So it's going to move up and down through the xylem primarily. So if you put it on as a foliar application, it will move into the roots, or if it does go on in furrow, again, it would have to be through an in-furrow liquid kit. It will go into the roots and then move up into the leaves. And then what happens is the bacteria is going to enter the plant cell. So this rectangle here is demonstrating a plant cell. And then it forms this little circle. And this is the vesicle that we touched on earlier. So it's inside this vesicle where the nitrogen fixation is going to occur. So that's kind of the best way to think of it, is that this vesicle mimics a nodule that we're used to seeing with pulse crops and rhizobia. So the one downfall to this, you can't see the Envita working. So not like with peas or lentils, you can pull them up and see the nodules there. You can't do that with Envita. The upside is the nitrogen is fixed right inside the plant cell, so it's made directly available where the plant needs it. So this is getting a little bit more in depth in terms of what's happening with Envita. So the first thing to know is that it is a symbiotic relationship. So the plant will provide a little bit of sugar. It needs carbon or in the form of sugar into the vesicle. And that's what it uses for energy. So nitrogen fixation is a very energy intensive process, just as it is with the Haber-Bosch from an industrial standpoint. So the plant provides a little bit of energy. It uses that energy, and then the nitrogenase enzyme, as I touched on before, and then it'll take that atmospheric nitrogen and convert it into ammonia. So sugar goes in, atmospheric nitrogen goes in, and the nitrogenase enzyme converts it into ammonia. And then the ammonia is fed back right into the plant cell, where it can be used for photosynthesis or building amino acids or whatever it's it for. So these are some pictures that were taken over the years while they were doing their R&D. So what this is demonstrating, in this case, the Envita was applied as an in-furrow application. So these are the roots of a corn plant and the leaves of a corn plant from the same plant. The bacteria was stained. So that's the little dark blue is the stained bacteria. And so demonstrating that the bacteria does enter the plant, it does enter the plant cells, as you can see it in there. And because it was an in-furrow application, it also demonstrates that it was systemic, because we can see it inside the plant cells of the leaves as well. So just a little bit of the work that was done to kind of figure out how it works and then demonstrate that it is working. In terms of proving nitrogen fixation, what the researchers did is they took all the same studies that they used for rhizobia and applied them to Envita. So there's a number of different ways to kind of demonstrate nitrogen fixation. We'll take a quick look here at a labeled nitrogen study. So the way a labeled nitrogen study will work is you label nitrogen using isotopes. And when you label atmospheric nitrogen using isotopes, it'll show up pink under a microscope. So it's a way of following atmospheric nitrogen. So they'll take that labeled atmospheric nitrogen and add it to a growth chamber, in this case with corn, and then just let the corn grow in that growth chamber. Once the corn is developed, they can then take a look at it under the microscope and see what's happening. So this is a picture here on the right-hand side of atmospheric nitrogen, because it's showing up as pink, being fixed inside the plant cell. So atmospheric nitrogen has now made it into the plant cell and is actively being fixed by the bacteria into ammonium. This third picture is now showing, again, showing up pink because it was originally atmospheric nitrogen, but now it's in the form of ammonium and it's being used by the chloroplasts for photosynthesis. So it demonstrates that not only does the bacteria fix the nitrogen, but that nitrogen is then used by the plant for growth. So we'll move in here to a little bit of yield information. I'll start off with this. This is a little bit older, so this is some work that was done in the US a few years ago, and it was nitrogen fertility rate study. And when they looked at it in corn in the US Midwest, what you can see is that regardless of how much additional nitrogen was added from a fertilizer standpoint, Envita always had a bit of a yield boost. So it kind of demonstrated that add your normal amount of nitrogen and then add Envita on top of that, and it was going to kind of elevate the yield potential of the corn. And that's still true today in corn. So that works really well for corn. We'll take a look at some wheat and canola studies, and I'll kind of show you why that might not be the case so much for wheat and canola, but for corn, it's still holding true. So this is some work. Again, this is trials out of the US. You can see mostly in the US Midwest here. So this is 2022 trials, very good response rate, 85% with just under 10 bushels an acre average across all these trials. So the results in corn have been very consistent all the way from 2019 when it was first launched. This is the overall trial results from 2019. So again, showing you that overall we're getting over 80% win rate and over 8 bushels an acre across all these trials. So in corn, it's been very, very consistent. Part of the reason I think is because it's really hard to over fertilize corn. It needs a lot of nitrogen. So Envita is always able to provide that extra boost. A few observations from corn. Typically, what we're really seeing, we can occasionally see an increase in cob size, but where we're really seeing the yield response from is the reduction in tip back. So instead of having less kernels make it to maturity, Envita helps more kernels make it all the way to maturity. And that tends to be where we see the biggest yield impact from Envita. So we're not getting bigger kernels, so the 1,000 kernel weights are always pretty much the same, but what we're seeing is more kernels making it to maturity, and that's leading to the higher yield. The other interesting component that we're seeing a lot of is actually very consistently is Envita brings the corn to maturity faster, which is a little bit counterintuitive to how nitrogen usually behaves. But our thought process is that if the Envita is in the plant, always kind of feeding that nitrogen there, there's no kind of stall or delay in growth. So it just helps it make it to maturity faster. Kind of a hypothesis, but that's what we think is happening there. Soybean trials. So again, this is 2022 data, so the more recent data. Really good results with soybeans in 2022. So 90% win rate and just under 5 bushels an acre. So this does bring up the question is, can you add Envita, will Envita work with rhizobia? And clearly the answer is yes. All these soybeans would have had rhizobia applied as well. So it does work with rhizobia. So it's not like nitrogen, and you can't use nitrogen with rhizobia because it makes the plants quote unquote lazy. That doesn't occur with Envita when you're using it with rhizobia. And this would be the long-term trial results from soybeans. So 70% win rate and 4 bushels an acre average in soybeans. Moving over here into spring wheat, typically didn't see a lot of visual differences in the field when looking at Envita treated fields, but this was one field where we did see a bit of a height response from it. But like I say, that wasn't very typical, but it was just a nice photo opportunity here. In terms of results in wheat, certainly not as strong as they have been in corn. And I think this comes back to my earlier comment that corn just needs a lot of nitrogen, and it's not that you can't max it out, but it's not probably economical for most corn growers to throw so much corn at it that you've maxed out how much the corn can use. But I think what we're seeing in some crops in Western Canada is that it is possible. So here you can see, we did a bit of a timing trial here in this trial. So this was a small plot replicated trial out of Swan River, Manitoba. And you can see really good results. So even untreated, we went 92.5, and we did not see much of a yield response here. However, protein did go up pretty significantly. So I think what that's telling us is that nitrogen wasn't the limiting factor. So we got really good yield results. Nitrogen wasn't limiting. So the Envita being in the plant couldn't really add more to yield, but it did bump up the protein a little bit. So this is another small plot replicated trial that Patterson Grain did just outside Winnipeg. And here you can see the yields are much lower, and we did see a yield response in this case. Protein, however, remained more or less flat. So in this case, the Envita went to yield instead, and I think it's showing in this case there was potential. The nitrogen wasn't the limiting, or was a bit of a limiting factor in the untreated, so the Envita was able to increase yield here a little bit. So I think it's a little bit more nuanced for us in Western Canada in these kind of crops in terms of how much nitrogen we add and what kind of response we're going to get. It's not going to be as kind of clear cut as corn is. So same kind of story here in canola. Again, AgQuest did some small plot replicated trials in Saskatoon. Just west of Saskatoon, it was very dry this year again there, unfortunately for the guys out of there at that area. So we saw a bit of a response here out of the Envita, but nothing too dramatic. And I think here it's pretty clear to say that the lack of moisture was the problem. So Envita helped increase yield a little bit, but it can't make up for lack of rainfall in any great degree. And then on the flip side, we've got, again, New Era did a canola trial. Looking at the different timings, we did see a bit of a yield response here as well. I'm not entirely sure why the later shows up better. I think that's probably just kind of variance in the results. But seeing a little bit of a response here, but again, very good yield result, or untreated yield result. So nitrogen probably wasn't a major limiting factor here. But it does demonstrate that Envita did certainly have some impact in the crop. In terms of potatoes, not entirely sure if there's any potato growers on the call, but throw them in here. Potato results are more like corn, and I think for that same reason, potatoes need a lot of nitrogen, and it's kind of hard to over fertilize them from a yield response perspective. So these are all yields in ton per acre. So again, really good results across these trials. These are not small plot replicated, they're just demonstration trials. Pretty much all out of southern Alberta, showing just under 3 ton per acre average overall. So far, I don't have all my trial results back yet from this year, but based on what I'm seeing, I think my focus for next year is going to be looking at comparing different nitrogen fertility rates. So if we reduce nitrogen fertility by 10, 20, or 30%, what kind of impact will Envita have there? And will it give us an opportunity to maybe reduce our fertility a little bit in either maintain our yield or even still give us a bit of a yield bump while we're reducing our nitrogen rates. So I think that's what I'm going to focus on in Western Canada on crops like wheat and canola, just because we're not seeing that same consistency as we are in corn and soybeans. In terms of the application, so you can see the different application timings here. For 2022, we were kind of being a little bit more conservative and just trying to kind of put it mostly with herbicide timing. As you saw in some of the results there, some of the later timings are actually showing pretty good results. So we can maybe extend these a little bit, say even into fungicide timing for cereals or canola. But we do have, more data supporting the herbicide timing at this point. But either way, it seems to work quite well. In terms of water volume, we recommend 10 gallons. We'll get into tank mixing here right away in a little bit more detail. If you happen to be applying Envita on its own, so if you're doing a trial, say you just want to do it on its own, we do recommend adding a non-ionic surfactant. So these are tank mix best practices. So we are mixing a live bacteria with pesticides. So we are trying to keep that environment as safe as possible for the bacteria. So our suggestions are try to keep your water volume at 10 gallons, if at all possible. I realize that saying 10, most guys think, well, that means 7.5, but the more water, the better, obviously, just to give that more dilution in the tank. Somewhat simpler tank mixes. So for instance, if you're doing it on wheat, say a grass, a broadleaf herbicide with the Envita, and try not to put in too much more other than that. And then add the Envita to the tank last, just so it limits the amount of time it's in that solution, and then spray it as soon as possible. So don't mix up your sprayer, mix solution, go have lunch and come back out and spray it after. Just try to get it in the tank, spray it out so it can get into the plant and survive that way. In terms of water quality, no known issues there at all, including if you're using a municipal water source, if it has a little bit of chlorine in it, that's not a concern. We've done a lot of testing there. We have done, I should go back to, I guess, tank mixing as well. We have done a lot of testing with some products, glyphosate being a big one. So we've looked at mixing Envita with glyphosate, and there's absolutely no concerns there at all. So that's a very safe tank mix for sure. And rain fastness is 2 hours. It just needs a little bit of time to get inside the plant. So just try to avoid getting it washed off right after you spray it. But if it's in good growing conditions, the bacteria should get into the plant very quickly.
Phil, would you like questions during the presentation, or would you
Yes,
rather wait to the end?
absolutely. No.
Okay, there is a question. Are these trials Envita's own, or are they independently run?
The small plot trials like New Era, AgQuest, those were all done by third-party small plot researchers. The potato trials would have been just demonstration trials by farmers, side-by-side trials. The corn and soybeans in the US would have been demonstration trials side-by-side by farmers. So all somewhat third-party, but they're not all small plot replicated. Some trials in there were just quote-unquote demo trials. So we do offer, for guys who are interested in using Envita, a performance guarantee. So the way the performance guarantee works is if you purchase a minimum of 1 case of Envita, and again, in 1 case will treat 160 acres, and then leave a check strip comparing treated to untreated. And if there's no return on investment, then we'll provide product to try again in 2024. So a couple of reasons why we're offering this program. One is we have done the lab work to demonstrate that Envita does work in wheat and canola, so it will colonize it and fix nitrogen in those crops, corn, soybeans, wheat, canola. But we may not necessarily see a return on investment. Part of it could be environmental conditions. Part of it could be maybe we accidentally killed the bacteria in the tank before it even got into the plant. And some of it, as I've kind of mentioned, is we're learning here as we go, and we fully understand and appreciate that. So guys that are using Envita early on are kind of helping us learn. And like we saw in some canola fields and wheat fields this year, if we're putting on too much nitrogen, or really high rates of nitrogen, the Envita may not provide a big response. So the performance guarantee just helps. And, you know, based on what we're learning, we'll kind of come up with a new idea on how to use it, and it'll give that guy an opportunity to do it again with the new information the following season. So these would be your numbers, your return on investment numbers that we've set in place for 2023. Just a review here of the product. So again, it's a liquid. Most of it goes on with a herbicide. It can go on in furrow if you have an in-furrow liquid kit. It would need to be cut with about 2.5 gallons of water or so, again, just to make it a bit of a safer environment with whatever else you're putting in that liquid in furrow kit. For Southern Alberta, I feel like that would be a feasible option. Envita the bacteria can't live on its own for very long. So when you put it on in furrow, you need to be pretty comfortable that the crop's going to germinate and get roots out pretty quick and into the in furrow, so that the bacteria can enter the plant and survive that way, because it can't survive indefinitely in the soil. So that's my talk for today. There was the one question. I don't know if there's any other burning questions.
All right, so we are ready for questions. I guess I will start it off while you guys think of your questions. Feel free to either, again, type them into chat, turn on your camera, or just use your audio. That's great. So Phil, when you're looking at the economic analysis, have you done any of those calculations as far as what you're anticipating in yield returns versus what the product costs?
Well, the product's around $16 an acre, and well, like in corn, it's pretty good, right? Because we're at 9 bushels. Kind of, if you go back to the, I can go back there, I guess, to the performance guarantee. These are kind of what we see as the break-even points, right? So in terms of wheat, canola, and that's based on roughly what today's commodity prices are set at. So we tried to tie these to commodity prices and the suggested pricing for Envita.
George, you have a question?
Yes, thank you, Jamie. Thanks, Phil. A couple of questions, actually. And I think you kind of alluded to it, but do you get a better response by foliar application rather than in-furrow application?
Oh, that's a very good question. What we're seeing, again, I'm going back to corn in the US because we've got a lot more data there. Where we're seeing there is no difference in terms of efficacy, whether it goes on in furrow or foliar. My concern for us in Western Canada, and again, maybe not so much for you guys in Southern Alberta, is if I put it in furrow and it gets really cool and dry and it takes 3 weeks for the crop to germinate, the bacteria will probably have died in the furrow. Whereas in the US Midwest, you know, that's like less likely to happen. And quite frankly, in Southern Alberta, it's less likely to happen. But that's my one watch out with the in-furrow application for us in Western Canada. That was a very good question.
No, it does seem like the foliar application is much easier than in-furrow anyway. So
yeah, the ease of use for us. Yeah.
And the other another question is, how long have you figured out how long it takes before after you apply the Envita until you actually starting to see an actual nitrogen fixation? You know,
I understand the question. Yeah, how many? It's probably I say a week to be a little bit safe. It's probably a little bit quicker than that, but yeah, within a week the bacteria should be in there colonized and starting to fix nitrogen.
And the last one isn't really a question, it's more a comment about your performance guarantee. Getting your product replaced isn't really help you any, because if it doesn't work the first time, because obviously you think it's going to work the second time then.
Well, that's, yeah, like what I'm saying. Well, first of all, it's a guarantee in case it's a live bacteria, right? So if it gets mishandled at some point, it might be dead and you don't even know it, right? The problem we have with Envita, and we're all working on a test to show that guys can do that, shows it's working, but it's not like rhizobia where you pull the plant and you can see the nodule. So when you apply it, there's no way to know if it's actually working. So it's to protect against maybe the product died before it even got in the plant. And like I said, we're learning as we go and we're fully acknowledging that. And so we're taking a little bit of the risk from the early adopters and saying, look, you're helping us learn. And if we like we're finding out maybe in canola and wheat this year, if you put a lot of nitrogen down, the results aren't going to be like we're seeing in corn. So maybe what we need to start looking at is cutting back a little bit or something like that. So it's because you're helping us learn, we can use those new discoveries and apply them to the following season. That's kind of our mindset there.
Thanks, Phil.
All right, we have a few questions in the chat. Mike is wondering, does the bacteria multiply once it's in the plant, or the rate that you apply, that's what you get?
No, it does absolutely grow inside the plant. So colonize, when I say colonizing, I could have maybe explained that better, but colonizing means bacteria grows inside the plant. So it will grow in the plant. And if you put it on at that herbicide timing, that 4 to 5 leaf stage, and the plant puts out new leaves, of course, the bacteria will move because it's systemic and grow into the new leaves as well. So it grows with the plant is the best way to think of it, which is why there's probably not a lot of difference in efficacy whether it goes on in furrow or foliar, because once the bacteria is in the plant, it's in the plant.
All right, we have the next question. Has Envita been tested on field peas?
Very little. So I guess the short answer is no. I had a couple guys do some quick sprays this year. I haven't heard back from them, but I'll only get 1 or 2 data points on peas. So I can't speak to what to expect there necessarily. Again, it'll work with the rhizobia. So, but I just don't know what kind of yield response to expect on peas or lentils, unfortunately. We really focused on wheat and canola here for year 1.
All right. And also, where do you see the best fit in Southern Alberta, dryland, irrigation, greenhouse forages, other crops? And I'm going to add to this question, are you going to be conducting any research in Southern Alberta this coming season?
So best fit, I guess, obviously, I guess under irrigation, you would eliminate the risk of having a really dry season. So that would help a little bit. But I mean, 99% of our fields are dry land right across North America. In terms of crops, I think what we're seeing is certainly corn is very responsive. Potatoes have been very responsive. I think as I mentioned, we're going to start focusing a little bit more on cereals and canola and seeing maybe if we cut back the nitrogen a little bit, if that'll give us more consistent results there. Presumably it will, but we haven't really done a lot of that nitrogen fertility rate trials at all in crops outside of corn. I feel like there's another question I'm forgetting.
Just if you were planning on any research trials in Southern Alberta this coming season.
I haven't crossed that bridge yet, but we're certainly going to want to do some larger scale trials. I'm not 100% sure if I want to do small plot trials, especially with nitrogen rate reductions. I just feel like bigger areas might be a bit better for those kind of trials. But yeah, we'll definitely try to do some demonstration trials across the prairies, but I'm not sure on replicated trials yet.
Another question on the Feedlit platform, how does yield compare between Envita versus a late season UAN application?
Oh, that's a very good question. I don't have a lot of top dress versus Envita trial results yet. I did ask a few growers to do it, and I don't have data back yet. Unfortunately, for reasons I don't really know, I've only got about half my trial results back right now. So I'm still waiting on a fair bit, but yeah, that's a very good question. And I'm not confident to answer that yet, unfortunately.
All right, no worries. And another question, is there any potential disadvantages to using Envita?
No, in terms of it can't hurt the crop. It can't do any damage to the crop. The worst case scenario is it doesn't, maybe the bacteria died and we don't know it, so it won't be able to work. But it's a common question because it is a symbiotic relationship. So people wonder if it's going to steal sugar or energy from the plant to the plant's detriment. And it's symbiotic. So the plant will only give the bacteria energy if it needs nitrogen and if it's healthy. And it won't put itself in a bad position by giving away energy if it can't afford to. So that's not a concern.
Ken, you have your hand up.
All right, Phil, can you hear me?
Yes, I can.
Thanks for the presentation. We're interested in hearing about the product as it's kind of gained global attention, and I think in particular due to the desired reduction in nitrogenous fertilizers and greenhouse gas emissions, which you know, kind of puts it in a little bit of a tough spot when expectations are sort of set. So I'm wondering, in particular, sort of along the lines more of developing sort of science-based data within Canada with stats and everything. I'm a little bit concerned about the approach at only field scale trials. A lot of times we've found in our work in the past that, you know, a difference of a bushel or two is not within the strength of a field scale test to be able to determine with confidence that those results are real. So I guess maybe I'm asking you a little bit more about the strategy in ensuring that, you know, where does it work, when does it work, under what conditions, for what crops. And I'm sure that's probably your next big job in your role, but Jamie sort of asked it. I'm just hoping maybe you could give me a little more details on why the focus on field scale and not small plot.
Well, because I've had conversations with other small plot trial researchers, and when I asked about doing fertility rate trials, is they couldn't guarantee me that where their block of land where they were going to do the trials, that they really understood if the nitrogen in that area was consistent, right? And then if we can't, this is how it was explained to me, if we can't guarantee that the nitrogen in that block is consistent, then how do you do a nitrogen reduction trial, right? So it could be based on past trials that were done in those areas that they can't provide that kind of level of confidence, I guess. But that's where it's kind of stemming from, is just some guys are a little bit hesitant to maybe commit to doing a reduction trial when they don't really know. But I don't know, you might have some other thoughts on that.
Yeah, I think I do. I think when we're talking about distinguishing small difference, this is more suited for small plot trials than field scale trials. And I don't know if Lewis is on the call, you can maybe comment on it, but you know, it's just was curious as to why you guys were taking that approach. Thanks.
Is Lewis on the call and want to chime in? I'll take that as a no. Okay, there's another question in the chat. How do plants take the bacteria from the foliar application?
So when it goes on as a foliar application, it's going to land on the leaf and form what's called a biofilm. And that's kind of how it attaches itself to the plant. And then from there, it'll either enter through the stomata if the stomata are open. And then once the bacteria get into the stomata, then it can move within the plant. Or if the stomata happen to be closed, there are some bacterial enzymes that are part of the formulation of the bacteria. And those enzymes will kind of help the bacteria enter the plant, kind of make its way in that way as well. Not unlike like a surfactant does for a herbicide.
All right. Are there any more questions that have come to anyone's mind? I see somebody's hand up. Scott, go ahead.
Oh, just a moment here. There we go. You can hear me?
Yes, I can.
Okay. Yeah. Yeah. Just a kind of something I was just thinking of was, do you think there'll be a fit with the emissions reductions? Do you think they're instead of like, so instead of a yield increase, you might be able to say emissions reduction because you're either using less nitrogen or you can prove that there's less emissions from nitrogen?
Yeah, that's part of the reason we want to do some of those fertility rate trials as well, is it kind of leads into that as well, right? If we can demonstrate that a farm can reduce their nitrogen by say 10% or whatever that number is to be, like keep the yield flat but reduce their nitrogen by that amount, there'd be a benefit to that as well. And you know, with the 30% nitrogen emission reduction requirements, like Envita can be a tool to help compensate for some of that as well. So that's part of the driving force behind wanting to do some of the fertility rate trials as well.
Okay. Yeah.
All right. Any final questions for Phil? All right. I will throw up the QR code for anybody. Nope, I won't. I lied. We got denied on this session. Yeah, CCA, sorry. um, so if there's any QR codes that you have missed along the way, make sure to let me know so that I can ensure you get the proper credits. If you are a pesticide applicator, don't forget to send me your information so that I can ensure you get your credit for the herbicide resistant presentation. And we will get a little bit of a break here before our next session will begin at 4 o'clock. So grab yourself a washroom break. We've been pushing you guys pretty hard today. We've had some excellent speakers, so I want to thank Phil for his time as well, and thank our sponsor, Alberta Canola, for their support for this event as well. So I will see you all back at 4 o'clock.